All right, it is time for the last part of learning about signal transduction. And that's learning about the last phase, which is the response of the cell. And this is kind of the goal of any signal transduction pathway, is to make the cell do something. So, we first talked about reception of the signal, transduction of the signal into the cell, and then now we're going to talk about the response, what the cell does. So cell signaling leads to regulation of transcription or cytoplasmic activities. So kind of two main types of responses. And the cell's response is sometimes called the output response. So ultimately, the end game of a signal transduction pathway is regulation of one or more cellular activities. Okay. This response, the cellular activities may occur in the cytoplasm or in the nucleus. Okay. The majority of cell signaling pathways regulate synthesis of enzymes or other proteins. So they regulate genes, basically. By turning genes on or off in the nucleus. So if this is the case, if this is what you want to do, regulate gene activity, almost always the final molecule in the signal transduction pathway functions as a transcription factor. And what a transcription factor does is turn on a gene. Okay, so let's look at that. So here we have reception of the response, transduction of the response through a phosphorylation or a kinase cascade, and that will activate some kind of kinase at the end of the cascade that will go into the nucleus. Okay, take an inactive transcription factor, phosphorylate it. Remember, adding a phosphate makes things more reactive. That changes its form. Notice this has a different shape than this, which of course changes its function. Now, in the inactive form, it can't bind to DNA, but in the active form, it can bind to DNA. And then the response is it turns on a gene, makes an mRNA, that goes out to a ribosome, makes a protein, and voila, we have our response. Okay, so that's one major thing that signal transduction pathways do, is activate genes. Another thing that signal transduction pathways do is to regulate the activity of enzymes rather than the synthesis of enzymes. So rather than make a new enzyme or make a new protein, it takes one that already exists and regulates the activity, either turns it on or turns it off. Okay, so let's have a look at that. Here again is our reception transduction pathway, and you see here's my kinase cascade, there's my G protein, there's my adenylate cyclase, my cyclic AMP, and then that activates protein kinase A, another kinase, and then finally we have an active kinase here, and it phosphorylates glycogen, okay, and turns it into glucose 1 phosphate, and so that is not what I meant to do. This will release glucose for metabolism. And that's the result. That's the response that we wanted from the cell. Okay. And I just want you to look here because I'm going to talk about amplification of a signal in a second. Let me clean this up a little bit. Okay. So if you look here, as you go down the phosphorylation cascade, the number of molecules activated at each phase increases, okay? Until you get a much higher, like 10 to the 6th molecules is a lot greater than 10 to the 2nd molecules. That's 10 to the 2nd molecules. Okay, so when I talk about amplification of the signal, I'm going to refer back to this slide. So the number of active molecules you can get in each successive step increases significantly. So the signal can get greater. So, you know, I alluded to this briefly in a previous screencast, but just a very few signal molecules, reception of just a few ligands can cause an enormous response in the cell because of the amplification of the signal. Okay. All right. So what other things can signaling pathways do? So signaling pathways can affect the behavior of a cell. It can change the shape of a cell. And I'm not going to get into this a ton. But what this signal transduction pathway does with mating factor is it changes the shape of the cell. So it just takes kind of a regular round cell and makes these schmooze, or these projections, like the red projections are the schmooze. And then that helps cells to mate. So it can change the entire shape of the cell as a result of the signal transduction pathway. So you don't worry about the details of this. But I mean, if you want to pause and kind of look through this, again, it is just a kind of a logical sequence with a different response from the cell. All right, so let's look a little bit at fine-tuning the response, making the response as perfect as we can, basically. And there are four main aspects to fine-tuning the response. One is amplifying the signal, which is what I just kind of talked about, and therefore the response. The response is much greater than the signal. Two is the specificity of the response, making sure you get the exact response that you want to. Three is the efficiency of the response. Okay, this can be enhanced by scaffolding proteins. This is basically like an organization to make it easier, make the response easier to get. And four is termination. And no matter what the response is, we always have to have a way to stop it, to turn it off. For example, the previous slide, those mating factors that change the shape of the cell, we don't want the cells to mate forever. At some point, they need to stop and go on and do their normal job. So we always need to be able to terminate the signal or shut something off. And this is kind of just a general tidbit that is true of every pathway. We have to be able to control the pathway and whether it's on and off. All right. So how do we amplify the signal? And I kind of talked about this. So our phosphorylation cascades, our kinase cascade during transduction can amplify the cell's response. At each step, the number of activated products is much greater than the preceding step. Remember, we had 10 to the 2, and then a couple steps later, we had 10 to the 4th. A couple steps later, we had 10 to the 5th. A couple steps later, we had 10 to the 6th. And again, this is, you know, the end result here is much, much greater than what we started with. Okay. Specificity of cell signaling or coordination of the response. So different cells have different collections of proteins inside of them that will coordinate these signal transduction pathways. And because there are kind of different groups of proteins, this allows the cells to detect and respond to different signals. Okay, not every single signal is going to be a mating factor. Not every single signal is going to be to initiate, you know, the production of a certain enzyme. So there are many, many, many different signals that the cell needs to respond to. So it has a different organization within it in order to do that. So, and even the same signal can have different effects in cells with different proteins. If you put a mating factor or a specific ligand on one cell, it'll have a different response than if you put it on a different cell. The signal transduction pathways can also branch and crosstalk or talk to each other to help coordinate signals. So let's take a little look at this. So we can have a signal molecule and go through some relay molecules and cascade and get a certain response. That same signal molecule with a different collection of proteins. So we activate a different protein here that can, and here's also an example of branch. With this different collection of proteins, so this protein is different from this protein, you can generate different responses, okay? And so let's go on here. This is an example of crosstalk pathways talking to each other. So you'll have that same ligand will activate a certain response, but if you coordinate it with a second ligand, these pathways can talk to each other. It can shut this pathway off or turn it on and get yet a different response. And then if you have a different receptor altogether, the same ligand can generate even a different response. Okay? And this just kind of, is just different ways that you can organize the responses in different cells. All right, efficiency, getting organized. So scaffolding proteins are used to kind of group proteins together. So it's a very large protein, and then other relay proteins are attached. So a couple of the inactive kinases in a kinase cascade can all get kind of stuck onto the scaffolding protein to basically get them closer together. So you can activate them all almost simultaneously. Okay, so this can increase the signal transduction efficiency by grouping together different proteins involved in the same pathways. Okay, and they can also help activate some of the relay proteins. So if we have our ligand binding to receptor, we have this scaffolding protein, and it collects, a kinase 1, kinase 2, and kinase 3 all together. So it's easier, this one activates this one. It doesn't have to go find the next one to activate. They're just right there. So it just makes it quicker, basically, and more efficient. All right, last, we need to be able to terminate the signal. So inactivation mechanisms are essential. It's super important to be able to shut these processes down, okay? A lot of this is done when the concentration of the ligand decreases. It binds to fewer receptors, and then the receptors revert to an inactive state and can shut down the signal. And that's it. The end. And that, my friends, is signal transduction. Okay? Remember the 3 steps: reception, transduction, and response. Okay.